All right, so we are now being recorded and I will begin my presentation. All right, so in this third video teleconference, I would like to ask again from anyone who wants to share. Okay, maybe we don't have to call each group, but anyone at all who wants to share on how they are in the group project. Have they found a way to solve the problem that was given, or do they need extra time? Okay, may I hear from you?
Hello, sir. Yes. Um, we have actually started now with our group project and kanang actually gamay na lang ang kulang sa mua na part, sir. And nala may question, sir, regarding sa to schematic diagram. Does it have to be present tense or in past tense, sir?
Thank you for that question. That is very relevant. In terms of your lab reports, let me talk first about lab reports. Since I already performed the experiment or the exercise, your schematic diagram and procedures should be in past tense because that already happened. But in terms of your group project, because you are planning a method to do in the future, it should be in future tense.
Sige, sir. Thank you, sir.
Okay, anyone else who has a question or wants to share about their group project? I hope in the methods that you've arrived with that you will not use and don't include materials or apparatuses that you don't even know the name of. Okay, the purpose is really for you to design a method considering the materials and apparatuses that you know. So for example, the light microscope, the micropipette. Okay, so um, the micrometers, those are the things that we know. And if you know about those and how they are used, I believe that's already adequate for each group to solve the group project. I also have maybe a good news for all of you on the group project. The deadline has been moved. Instead of at the end of this week, the deadline for the group project is now going to be next week on January 29. So you have ample time to really design your method. Okay, so that will be the new deadline, January 29 for the group project. Any other concerns on the project? I think with my announcement, many of your concerns were erased right away. You have enough time. Okay, so all you need to worry about right now is the deadline at the end of the week is for exercise 3 on the hemocytometer. And please also don't wait for next week before you really work on the group project, because there's another exercise that will coincide with the deadline of the group project next weekend. Okay, I think we've dwelt enough on that. And if you have questions, specific questions, you can always message me through eLearn or through Messenger. So just a reminder, this is the title page, and I believe all of you followed the format of the title page. I also do not encourage you to reuse the lab exercise itself, the manual. So for example, I think there is a group who used the format or the manual itself with the text in the background, and then they just filled in the table. That's not really wrong, but I encourage you to create your own table from scratch in the next lab report that you will be making. And I've already returned your first lab report just a while ago. And just to reiterate the criteria, first, objectives. Objectives have to be specific, okay? So do not give objectives that you yourselves would be wondering, what am I, what is my goal for this exercise? So for example, if your goal is determine the accuracy of the micropipettes, okay? then state just that. Be specific. Okay. Do not make an objective that to learn. Okay, because when you learn, there are many things that you can do to learn. So be specific in your objectives. Okay. Next, for the schematic diagram, because the exercises have already been done, so they have to be in past tense. They don't have to be in complete sentences. They can be in phrases, but in past tense. And since this is a schematic diagram, I am expecting a flowchart-like format. Okay, and I think all of you were able to make a make something of a version similar to a flowchart. Okay, that was fine, and don't make it too long. Okay, it should be concise. That's for the schematic diagram. For the results tables and figures, many groups went extra mile to present figures on the parts of the micropipette or other tables, no, aside from the tables that were required in the lab report. There is no problem on that. However, my comment is, remember I gave you raw data on the before and after masses of the second and third trials. Okay, I gave you a table. There were groups who also presented that still in their lab report. And my opinion for that is that's already data in the background. That's not the main or the final data. Those are not the final data that you need to present. So in the future exercises, just present the data that are already final. Okay, so that you won't also have an overly long laboratory report with a lot of tables. For the discussion, it should be, it should explain the results. Okay. There were some instances when the discussion was long. Okay, it was written beautifully. However, you only devoted one paragraph to discuss the results, and you didn't even tie loose ends on the results. Okay, you simply mentioned it and then you didn't discuss or analyze anymore. So for the discussion, it should be majority an analysis and a discussion regarding the result, the actual result of the exercise. The generalization is the conclusion, and it should be based on your objective. So this is where the problem on the objective is magnified. If your objective is not specific, your generalization will also be problematic. Okay? Because you don't know what to answer because your objective is so broad. So make it very specific so that when you reach your generalization, you also have specific answers. No problems on references so far. And writing quality is about grammar, of course, capitalization, complete sentences, etc. So I hope you will be able to view the corrected lab reports that I've sent through eLearn as well. Any concerns though about this one? Okay, just to reiterate, for the group project, it should be future tense, not past tense. So for our schedule, we are now on the 22nd, not on the 21st, but we have moved activities a bit, but we are not late because we just exchanged several of the activities. We are on exercise 3 right now, and next week we will be doing exercise 4, okay, as indicated in this calendar. However, it won't be exercise 4 next week. It will be exercise 5, osmosis and the plasma membrane. So I'm informing you ahead right now. Maybe you want to read ahead for yourself. Read exercise 5, okay, which is osmosis and the plasma membrane. That will be the next exercise for next week. And as usual, we want everyone to have the opportunity to test themselves on how well versed they are on the exercises that were presented through the videos. Okay, So this is our self-quiz number 3 on exercise 3, the cell counts using the hemocytometer. And I encourage everyone to answer. Okay? Be enthusiastic, answer. Sort of compete with yourself. Maybe compete healthily with others. Paunahan kayo. That's fine as long as it's healthy competition. Okay, the goal is for everyone to learn from this self-quiz. If there are items in the self-quiz that you yourself do not know the answer of, please take note of that so that you can go back to it in the lab manual later on. Oh, are you ready? Let me hear from you. Are you ready for self-quiz number 3, which is non-graded? Anyone? Seems so quiet. Yes, they're ready. They messaged. Okay. So I hope when you answer through your audio because I'm presenting the slide. I will not be seeing your text messages. All right. So number 1, how many counting chambers does one hemocytometer have?
2.
2, sir.
2, sir.
Exactly. Okay. So when you actually use a hemocytometer, you don't just use one counting chamber. You average the number of cells, the cell count in the 2 chambers. Now that's why that's part of the method in your exercise to average from the 2 chambers. Next, what cells were counted?
Yeast
Yeast
cells, sir.
cells, sir.
Both of you are correct. Are yeast cells prokaryotic or eukaryotic?
Prokaryotic.
Eukaryotic, sir.
Eukaryotic. Like,
Make up your
eukaryotic.
mind.
Eukaryotics are simple.
Eukaryotic. That's correct. In fact, it's the model organism, remember, for single-celled eukaryotes. Okay, that's your yeast cell. So we use Saccharomyces cerevisiae for our specimen in this exercise. Next, what stain was used?
Methylene blue.
Methylene blue. How many percent of the methylene blue was the solution used?
0.01%.
Yes, okay, 0.01% of the methylene blue solution. All right. Next, what color should the dead cells be after staining?
Blue.
Blue. Are you sure?
Yes, sir.
Ah, okay. We won't answer why because you are still making your lab reports. Okay, we will not answer. But I hope you already know the answer why the dead cells will be blue and the living cells will be colorless. So I hope you can answer that in your lab reports. All right. Next. So this is a counting chamber. Okay, this is one of the counting chambers. So question number 5 is, what is the surface area of the blue square?
Um, 3 cubic millimeters, sir.
Surface
Oh,
area,
sir.
meaning to say two-dimensional.
9 mm squared, sir.
9 mm squared. Can you tell us how you computed for that?
3 times 3, sir.
All right, the dimension of this square is 3 by 3. So that's 9 square millimeters. Okay, don't forget the unit, it's square. Okay, next, what about the red square here?
1 mm squared, sir.
How did you compute for 1 mm squared?
1 times 1 mm, sir.
All right, so this is 1/3 of the dimension of the blue square. So the blue square is 3, therefore one dimension of this is 1. So 1 times 1 mm will be 1 mm squared or 1 square millimeters. Okay, my slide seems to be having its own life. Okay, this red square is called, how do we call this red square?
Quadrant, sir. Bigger quadrant.
Yes, that's the quadrant. And we can say it's the bigger quadrant. Number 7, what's the area of that green square? It's also magnified here on the right. You can see the small quadrant on the right. You may use your calculators or cell phones. Has anyone answered? Still calculating?
Not sure, sir, pero 0.2 times 0.2. 0.04 millimeter square? No.
How did you get that?
Patong, ang isa kas, ay, sure. Times ang 0.2 sa 0.2, sir.
How did you get 0.2?
Gi-divide ang ganing ang 1 millimeter to 1, 2, 3, 4, 5 squares.
All right. Okay, so you also have a different approach, but 0.2 is correct. I say you multiply that 0.05 4 times, right? Okay, you have 0.05 plus 0.05 plus 0.05 plus 0.05. That's 0.2. You're correct. And 0.2 times 0.2, as you mentioned, is 0.04 square millimeters. You are correct. That's the surface area of the small quadrant. Okay, so I really hope all of you will be able to remember these dimensions, no, or will recall how they are computed when you use the hemocytometer again soon. Okay, because as we know, the hemocytometer is also used to do blood counts. So in the field of medicine, there might be a time when you will be using a hemocytometer. So don't forget how the dimensions here are like. So we also know that when we put the sample at the center, when we put the the cover slip on the blue line here, there will actually be a gap between the upper surface of the hemocytometer and the inner surface of the cover slip, and that's 0.1 millimeters, okay, the one in red. So we are not talking here about 2 dimensions anymore. We can sort of make the counting chamber like this, 3-dimensional, okay? And we can sort of put a cube or a box on top of this. And the height of the box is 0.1 mm because that's the gap between the surface of the hemocytometer and the cover slip. So naturally, there will be volumes involved. So the next items will be about volumes. Number 8, what is the volume about above square blue square?
$27.
How did you compute for 27?
For me, sir, since it's shaped like a wait, never mind, never mind. I thought I was ano man good, like it's shaped as a cube, so ano na ko, by the power of 3 na ko, sir.
But you're close.
Okay, okay.
So Anyone wants to answer? Or you may answer again.
0.9 mm cube, sir.
All right. How did you solve for that?
3 times 3 times 0.1, sir.
That's correct. Okay, because the height, remember the gap between the cover slip and the hemocytometer is 0.1. So that will be the height of your square here. No, so it's not a perfect cube. So it's 3 times 3 times 0.1 or 0.9 cubic millimeters. All right. What about the red square?
0.1 mm cube, sir.
Okay, can you tell us how you solve for that?
1 mm times 1 mm times 0.1 mm na height, sir.
That is correct. 0.1 cubic millimeters for the red square or the large quadrant. What about the small quadrant? We will wait.
0.004
And the unit is?
millimeter cube, cubic millimeter.
That is correct. How did you solve for that?
Sir, gapon ganiya, sir. 0.2 times 0.2 times 0.1 millimeter.
All right. So those are the volumes on top of these squares. No, when you consider the 0.1 mm height or depth, the gap between the cover slip and the hemocytometer. So do you remember in your lab report, there's a portion there wherein you're supposed to calculate for cell density per ml? When you counted the yeast cells, what did you use? 8, 9, or 10? Look at the squares. Green, blue, or red?
Green.
Green, sir.
That's correct. You use the green one. Because if you use number 9, you will be counting thousands of them because they are so small. Okay, yeast cells are very small. So instead, we used HPO and we viewed it. We viewed the smaller quadrants, and that's the one that I gave to you, the pictures that you need to count. So take note, the volume on top of this square, as you mentioned just now, is 0.004 cubic millimeters. Remember when you needed to solve for cell density, what do you need to multiply, the last multiplier?
10 to the 4 to the power of 4, sir.
That's when you use the large quadrant, number 9. Okay, I hope you didn't do that in your lab manual. We
Quad is
used
sir,
small square.
2.5 times 10 to the fifth power.
Okay, or 250,000. Okay, so there is a reason for that number, and I asked you on the one of the videos that I posted why 250,000, and the answer behind that is the volume of this is 0.004 cubic millimeters, and you need to convert that into an ml because cell density is per ml. All right, so for the rest of the calculations, I leave them up to you. But I'm giving you sort of maybe a hint if you did not realize yet that it has something to do with 0.004 cubic millimeters. the 250,000 or 2.5 times 10 to the power of 5 multiplier for cell density. All right. This wraps up our self-quiz for exercise 3. If there were questions that you were not able to answer, you may review this video again because I am recording this and I will be posting this in eLearn later. All right, let's proceed. For the prelim project, as I already mentioned, the new deadline is now January 29. That's next week. And this coincides with the next exercise. So please don't wait for next week in order to start working on the prelim project again. Okay, do it now so that you will have ample time. And this Saturday is the deadline for exercise 3 hemocytometer, the one that we just talked about. Next week, instead of exercise 4, we will be doing exercise 5, osmosis and the plasma membrane. With that, do you have any concerns or questions? This is a short video teleconference. And I appreciate you sharing your thoughts and answering, because that's really the purpose of this. We cannot do face-to-face, but we can do voice-to-voice instead of you watching at a pre-recorded lecture. Any concerns at all regarding our subject?
Mag-ask lang ko sa schematic, sir. Kinahanglan nga na, sir, past tense.
Are you talking about the schematic diagram of the prelim project?
No, sir. Sa exercises.
Okay. For the exercises, because I already performed the exercises and you are looking at the results, they were performed in the past, so use past tense in your schematic diagram. But let me add, for the prelim project, which you're still planning, your schematic diagram should be in future tense for the prelim project.
Thank you, sir.
Okay, any other concerns? No concerns? If there are no concerns, then I thank you. I thank you in joining this video teleconference, no, despite perhaps whatever you're experiencing right now, hardships, etc., you still joined. And the recording of this will be posted later on. Tell your classmates. I'll see you on the next video teleconference, and have a productive day ahead. Okay, so you are now all dismissed.
Thank you, sir. Have a
you,
great
sir.
day.
So,
Sir, goodbye, sir.